Okay, so I'm going to try to, I'm going to invite you to interrupt me and ask questions, and if I don't go through all of these slides, that's fine. I don't care. The objective is to get you guys thinking and talking. Maybe we can, yeah, I'm sorry, we have to turn this light down on here. Make people do contradictory things.
Keep the board light on. Huh? Okay. No, there are many switches. Cannot use the switch.
Okay, so they're all together, that's fine. Maybe I'll ask you to turn it back up in a while, or maybe I'll just skip that part. So, all of you people who we call students who are actually young scientists, have you been studying for critical analysis? So you're ready? Okay, what's the big question for this whole meeting? There's no room for being shy in this profession. What's the big question? Say it again. Study. You have to put a question mark on the end of that. Why study axonal transport? That's an interesting question. Scott, why? That's right. And because it's there. And because watching things move is cool. Right? It really is. I mean, that's why I do it. I'm moderately curious about how the nervous system works, especially as my nervous system slows down and doesn't work so well anymore. But really the primary motivation for me for doing science is because I'm curious and I really enjoy thinking about biological machinery and how things work. And so for me, the big question here, see, I gave you a hint, no one was paying attention. How do things move? For me, that's the big question. And if you look at this man is moving at about 10 meters per second. Who is that? Usain Bolt, world's fastest man. Now over here, Scott was showing you some transport in axons. This is transport in axons. And what's the speed here? Scott told you what the speed is. What is it? 1 micron per second. Have fun. 1 micron per second. 10 meters per second. Do you believe me if I say that the machinery that does these two things is almost identical? You believe me? Oh, I find that kind of astounding, but it's just engineering, right? And the machinery is this. And so myosin drives muscle contraction, and the muscles are hooked up in fancy ways, and the myofibrils are put in series, etc. And so you can go 10 meters a second. Over here is another motor and filament combination, and it's not engineered in such a fancy way, and it goes at about 1 micron per second. I think that this is kind of cool. Anyway, what we're going to do now is I'm going to go along and talk about the structure of these motor domains. I'm not an expert in this field, but I think for the purposes of our class, I can generate questions and discussion. And how do we word this? Oh, okay. So, next point. You have the machinery for biological motion of this sort is motor and filament. Now, there's another way to move things. What is it? What? I can't hear you guys. Speak louder.
And we have to use the microphone that is there.
Just yell at me.
So diffusion is active transport.
Diffusion. Okay, diffusion can move things. Very good. But how about big things inside of a cell? I mean, inside of a cell, the viscosity is high, the scale is low, the Reynolds number is low, and so as soon as you stop pushing on something, it stops. And if you have a large object, a small object like a water molecule or even a moderate-sized chemical is going to be bounced around because it's getting bombarded by water that's flying around all the time, right? And so diffusion works great, but for large objects like the stuff that Scott's been, I'm going to get a feedback here. The stuff that Scott was talking about, the organelles, I've lost my train of thought. You can push it another way besides motors and filaments and besides diffusion. What is it? Polymerization. Polymerization of filaments. I think that probably is the original motor. I mean, beyond diffusion, right? It probably, you had to invent a filament first, right? And the filament can grow longer, it can shrink, and if you hook things to the ends in a clever way, you can move things that way. And then I think motors came later. So it's important to not become too obsessed with this model that motors move everything. Or I guess that the motors that we call motors are the only ones, because filaments are perfectly good motors too. But that's kind of an aside. So the point of this slide is sort of a rough gauge of scale. We have 3 different families of motors that Scott talked about. We have the smallest one, kinesin 1. Motor domain's only about 340 amino acids. The medium-sized one is myosin that walks on actin. It has 650 amino acid, sort of minimal force-producing domain. And then dynein is huge, 3,500. So each of these interact with a filament. These 2 interact with microtubules. The relationship between this scale and this scale isn't quite right, but it's pretty good. You can see the straight protofilament tracks along the microtubule. Here in the actin filament that the myosins walk on, you can see that the track is actually filament. So it's 2 strands of actin that are coiled around one another. All right, so it has a helical pitch to it. Most microtubules don't have very much helical pitch. They have straight protofilaments. All right, so I'm going to start out by talking about myosin. It's the motor that's been studied to good effect for the longest amount of time. Up here is another image, electron microscope image of a myosin 2, muscle myosin 2 dimer. And what it looks like, cartoon style, is a single heavy chain dimerizes with another single heavy chain with this long coiled coil. And up here is a globular domain at the end terminus. And associated with that are 2 light chains. One's a regulatory light chain and one's an essential light chain that helps the protein fold and helps control its structure. Okay, so the strategy for Usain Bolt's muscles is that you take a lot of these myosin 2 dimers and you polymerize them. And so they polymerize by going mostly in parallel, hooking their tails together. And so what one has is tails, head, another tail, head, and so you make these bipolar thick filaments. All right, so now that's the basic structure. Everybody's, I think, pretty familiar with this stuff. If you have a question at any point, just interrupt me and yell, because I don't hear very well. So getting to that 10 meters per second requires just biological engineering at many levels. The bipolar thick filament, this is the single unit of contraction in the myofibril. Bipolar thick filament, it's suspended between opposing actin thin filaments. And the myosin the myosin 2 walks toward the plus end of the actin filament, which is embedded over here. So it's walking from the minus end toward the plus end. This one's walking from the minus end toward the plus end. And what happens is that the sarcomere shortens. That's muscle contraction. Fair enough. Highly ordered, many myosins working together. Why is that important? Well, let's see. Am I going to talk about it? I think it's important to talk about it now. So when you think about this, it's not important for a myosin to be processive. Okay? So a myosin 2 in muscle can run. It doesn't have to walk. So the myosin can go down and it can hit the ground and leap, and its sibling myosins will keep it from drifting away from the thin filament, and they can, in their thousands, keep contact between the thick filament and thin filament. It doesn't matter if myosin is a nice processive motor and whether or not its partner in the dimer cares about the two heavy chains don't have to care about one another, right? All right. Yes.
Curious. If you have fewer biofilms, does that mean you contract slower?
No, I think the rate of contraction for the rate of movement of a single myosin is going to be the same as many. The thing that it will do is it will have more power, more force. And I guess under load it can slow down. So maybe you're right. Maybe with fewer myosins you would go slower. I don't know. Who's thinking about this? Rube?
Yeah, I agree with that. With fewer myosins you would probably go slower.
If you have load. Yeah. Yeah.
And in this case, load is what?
Load is the resistance to muscle change in structure. Or if you're picking up something, it will be the same.
It will be the tension in the muscle being used.
Right. Yes.
In this case, where multiple myosins are acting together to undergo contraction, shouldn't it be the case that they should coordinate with each other and know what one myosin is doing? So they should be in sync.
Why?
So otherwise, let's say if one myosin is taking one step and the other myosin is not taking the same step, it will not be able to contract as efficiently as it should.
But if you have 10,000 myosins all working together as a team, and let's say that 100 of those at any one moment are engaged in producing force, then the other, 10,000, 9,900, does it matter what they're doing? Or if they're going to kick in next, or can it just be stochastic as to which myosin engages with the thin filament?
I think also the point is that the ones which are not engaging, they're not impeding others. Because the contact time for these myosins is very small.
Is that what your point was?
Maybe that's what.
So you don't want to have drag, right? And so the way the system is engineered, or the way actually not the system, but the myosin motor domain, is that it actually engages with thin filament for a very brief instant of time. And then it gets off so that it doesn't create drag for all of its cousins.
But even in that case, with whichever myosins are interacting, they should all be in step, right?
That's an interesting question. I don't know. Maybe it also depends. I mean, it matters how much compliance there is, how much stretch there is in the system, because you can load up a rubber band asynchronously and still keep tension on it, right? I don't know how rubbery the thick filament is, but it has to have some finite extensibility.
You have to remember that both the myosin and the actin have a registration issue, because it's not a uniform binding surface, it's a series of discrete ratchets, if you will. so that the actin it can only bind the actin at certain points along the actin filament. So it sort of automatically becomes, you're only getting the ones binding that are in the right position to be able to bind at any given time. So the ones that are not in the right position, so there's almost a default way of getting a sync roughly. Only the ones that are in the right position can.
But adding on to that, if there is this in-step thing, if all of the myosins that are attaching in step, then wouldn't processivity be required? Because then the siblings are not healthy, right?
No, it doesn't matter. So,
We have a large geometric structure which even if you do part of it, there can be an post-generate a small part and in another place at a later part. It's not like a vesicle process.
if you were going to engineer it, if you were going to build the best contractile machine out of the system that you could, you would probably do it by making things synchronized so you have one event, the next event, and it's all organized for maximum efficiency. But what do you know about biological evolution that argues against that as a design plan? It's a series of accidents, right? And so you have an accidental change in a protein structure that makes things a little bit more efficient. Then you have another one. And they don't have to make sense. They just have to build on top of one another, making an improvement so that you can reproduce better. That's what it's all about.
But I think the synchrony will result in some kind of disadvantage. If
Some kind of what?
disadvantage.
Oh, disadvantage.
If the muscle is under the tension and all the muscle filaments are in the synchronized manner, then at a certain point, all will have to.
I understand. So I'm not saying all 10,000 would be engaged and do one step and then all 10,000 would come off. That doesn't make sense. But, so you're right. Okay. So, collectively, the thick filament is processive. But the myosin dimer, an individual dimer, is not processive.
I think that's the critical part. Processivity is a scalable topic. And so if you talk about it at the individual molecular level, is one thing. If you talk about it at the total filament level, is another thing. And
The filament is processive.
And part of the problem is that if you have it too synchronous, then anything that gets out of step can disrupt the whole machine. By having a swapping mechanism, so to speak, it's a much more robust system in which it doesn't go wrong with speed. And you can look at different muscles, like you compare insect flight muscle to, Bill's thigh muscle, and there's a huge difference in efficiency of contraction.
That's absolutely true.
The structure is so much more regular in insect flight.
In general, when we think of the synchrony, we generally think in the temporal manner of synchrony. But I think in this case, there is a positional control of the synchrony. Means whenever the motor domain of the myosin filament is at the right position, then it will bind and it will synchronize with all other which are at the right position, and others will be in detached conditions at the same time.
But they may not be the same size,
They-
the same enzymatic.
It may not be temporally. I mean, geometrically, it can be synchronous.
Physically bound yet, but they may not be at the same step before breaking down.
All right, we have to move on. Thank you for that discussion. Oh, how does it relax? How does the sarcomere relax? Well, the myosins stop engaging with the thin filament, and then there's an opposing muscle that can actually help the sarcomere. It contracts, and the muscle in question will lengthen, right, and it slides apart. So it's passive sliding apart. Is that answering your question? Good? Not good enough. Well, you can talk to me later. That's fine. Excellent. All right, so I'm going to step on now. So well, what?
Just age and sufficiency goes on decreasing with more.
Age?
Yeah.
With age?
Efficiency of contraction relaxation goes on decreasing with the age of the muscle.
I can assure you that that's true. That is a true statement. Now let's talk about how the myosin motor domain works, or at least how people think it works as of recently. And so Ivan Raymond, great triumph of structural biology, fought for decades to get crystal to form with a fragment of myosin, the S1 fragment, that is the motor, the force producing part. Got it to work, and he and his wife, Hazel Holden, and their collaborators got this atomic structure. Beautiful, beautiful work that was really, it's just a very impressive set of science that they accomplished. And they and others have since learned quite a bit about the structure and how the structure changes in different parts of the cycle of ATP, binding, hydrolysis, and product release, and how that cycle is probably coupled with the other cycle that's important for filament-based motors, which is bind the filament, conformational change, and then release the filament. And that's the way, you have to have the coupling of the two cycles. And so, let's see here. So, here is a cleft, right? It's a mouth in the head of the myosin, and this is where actin binds here. ATP binds in a cleft that comes in through this way. This is a light chain, this is a light chain, and the purple is the myosin, right? So the different colors here are just different domains of the myosin heavy chain. This is actually a light chain, this is a light chain. So, the light chains in this sort of conceptual scheme act to stiffen the alpha helix that is the myosin tail that goes off here, right? So it's an important mechanical stiffening. So what happens? Okay, so first, here is myosin 2 head bound to an actin filament. You notice that the cleft is closed, all right? And there's no ATP here. What does this bring to mind? Is this good? It's not good. That's what you're going to look like when you're dead. That's what your myosin is going to do when you're dead, because there's no ATP around. Your mitochondria have gotten tired. There's no ATP. No ATP in here. The thing's locked on to the thin filament, and you cannot change the length of your sarcomere. Okay, so add ATP. ATP comes into the active site, and when it comes in, the active site causes a conformational change in the actin binding site, opens it up, and now it doesn't bind to the filament very well. Okay, so the affinity of the myosin for the filament goes way down. And so what happens is it releases. So ATP binding causes release of the myosin from the actin. That's what Scott told us. All right. And now what happens? You have active site closes on the adenine ring. And so you have a closing of this cleft. And that encourages hydrolysis of the ATP. So the gamma phosphate gets clipped off, but it's still held. And what happens now is, look at what's happened. This stiff tail out here has changed its position. So it's like cocking a spring, all right? And so you have changed the conformation while you're off the filament, and now the phosphate release, wait a minute, I'm getting ahead of myself. So, contact with the proper binding site of a thin filament triggers the release of phosphate, and the release of phosphate causes closing of this cleft that increases the binding strength between the myosin and the actin, and it also causes, this is an imaginary intermediate, no one's ever actually seen this state, but what it causes is you go from this to this, boom, and you have inorganic phosphate release, and then you have ADP gets kicked out, and you're back to the original state. So what you have here is you have the change in conformation once you're bound to the filament, like from here down to here, it's 5 nanometers. Okay, so that protein has just gone through a 5 nanometer swing of its tail, or its stalk, whatever you want to call it. Does that raise any questions for you? 5 nanometers. So it made its 5 nanometer push toward the plus end of the actin filament. How far is it between binding sites on the actin filament? Say it again? 1 monomer? That's what I thought too, but you know what? That's wrong. What's a different answer? Come on, I gave you a hint a while ago. Root?
I'm trying to prompt it. There's a helical pitch.
Helical pitch means it turns like
3.14 angstrom? No. So this is actually 50 angstroms. It swings 50 angstroms, so 3.4 is very small. The helical pitch is the answer because it doesn't show very well here, but if you go back to this slide, you can see that there is a periodicity, so it's a twisted ribbon, right? And so if a myosin is fixed on this side, 180 degrees, it can bind here. It can't bind here. It can't bind there. It can bind here again. 36 nanometers to get from one binding site to another in the same geometry. All right? And so that's another reason why myosin 2 really, in a muscle, it probably should not be processive. Because if it's trying to get its partner dimer to the next binding site, how big of a step would it have to take? 36 nanometers, right? Okay, so it doesn't. It just goes down and it kicks 5 nanometers, and then its cousin over in the next country, or not in the next country, but its cousin on down the cable gives another kick, and there's 1,000 different cousins kicking along asynchronously, and they keep the machine going in one direction, but they don't need to be processive.
the non-muscle myosins also have to take pretty big steps.
They do.
And so do their structures differ from the muscle myosins in, say, not the motor head itself, but something else which allows them to take these big steps?
You understand this question. So muscle myosin works in a cooperative machine. So processivity isn't very important, but you have myosins like myosin 5, myosin 6 that are on organelles, and they have to work all by themselves, or probably they can work all by themselves. And so they have a severe problem to solve. So what do you suppose they do to solve the problem? Gents, if you guys don't give me answers, I'm going to call on you. Sonia, you don't like, let's see, I can't remember your name. Chewy? Okay, so what's the answer? Well, but it's easy. It has to be processive, right? We just went through the logic of what you need to be processive, and Sandhya just said so. So what is it? It's dimer part, so it's a dimer, right? It has to reach the next binding site. How far away is it? 36 nanometers. And so what does it mean about the way the thing works? It has to take a big step. Scott.
Unless it's following the pitch of the helix.
Unless it follows the pitch of the helix. And I have a vague memory of some in vitro assays where you do have it follow the pitch of the helix.
Myosin 1 does that.
Yeah. Myosin 1 is a tiny little protein, but I'm getting ahead, so let's go to Sanja's question. Oh, wait. I'm going to answer your question in just a second and show you the structure. But first, let's look at this, which is a cartoon now of how myosin works. Go ahead.
It is like A to B. So A to B is just binding of ATP hydrolysis there, right?
Correct. ATP binding causes release of the myosin from the actin.
So some work exactly with hydrolyzing the ATP.
The hydrolysis of the ATP causes cocking of the spring.
Hydrolysis is slow relative to release.
Sir, I have a question. Sir, if we are having some ATP, it is not available. What will happen in the muscle?
It will go to this state, right? It can't hydrolyze, so it can't do this active site cleft closure, and so it's not going to be able to interact with the actin filament strongly, and it's not going to be able to do its power stroke.
Also, changes present which would act like poisonous things because they can cause anything. There were heart muscles and all those things. They will stop once. Those inhibitors like ATP, we should not hydrolyze.
Would you state that again? I missed the beginning of what you said.
So what I need to say is there are such molecules like ATP-like molecules which could bind the myosin, but they are not hydrolyzable. All the such molecules present which act like poisons.
Are there poisons that act like an ADP? Is that what you said, or act like a non-hydrolyzable AT? Non-hydrolyzable
AMPP.
AMPPNP.
It causes a relaxation or a release of myosin from the
And that will definitely poison you, I suppose. I mean, has anybody made a mistake?
It doesn't cross membrane.
So none of these charged molecules cross membranes very well, but if you inject it into your heart muscle cell, then your heart muscle cell won't. Diffusion. Very important thing is diffusion. No. Well, I mean, that's a good idea, but it's the machine is much more complicated than you're thinking about. Okay, and so there's all these other parts to the myofibril that regulate whether or not the thing is going to contract. And what you're thinking about is how you sleep. And so there are troponin. Sorry, I don't even have troponin on here. I took it out. But this green filament that wraps around the actin, see that real thin green thing? That blocks the myosin binding site until it gets the right concentration of calcium, and then it rolls off of the actin binding site, or the myosin binding site, and then the myosin can engage. And so that's what controls the muscle contraction.
All embedded in there is the sarcoplasmic reticulum, which is
It's just this myofibril is wrapped up in a reticular membrane network that's chock full of calcium. And when an action potential fires along the muscle membrane and goes down the T-tubules and into the sarcoplasmic reticulum, and that releases the calcium that hits the troponin C, that rolls the tropomyosin, and then the actin can happily walk along the filament. AMP or AMP? Scott said 10 to 1.
Roughly, it's a much lower affinity. There are other ones, AMP, PCP, and some others that are out there. Which they vary. Different motors have different affinities for different animals.
So it's not a very good poison.
Right.
We'd have to load our heart muscle up.
For Myosin, it's a good one for kidneys.
Excellent.
But ATP gamma S is
And so the grand scheme here, this is a cartoon, and there's some structural reality here. You can see there's a globular part of the myosin head, and here's its stiff neck, I guess you could call it. Tail, I called it before. But you have, okay, phosphate release. Okay, now here comes ATP. It releases, hydrolyzes, changes conformation, binds, phosphate release, and that swings the neck.
what's not realistic here is that it's only one head that's acting.
Yeah, well, but that's part of the story here is that this is not processive because it's in a muscle. And the other head doesn't really matter. And I'm not sure of this, but I think they don't even care about each other, other than they help each other polymerize into the thick filament. They probably do care about each other. Okay, yes? No, it's random. I mean, you could alternate these heads or have them be in an asynchronous, random. I've looped the movie, or Sarah Rice and Ron Vale and Milligan looped the movie, so it looks like it's doing one step and then one step and one step, but that's just so that you can see it over and over again. That's not really a processive stepping process.
When it is taking one step as such, the next myosin is taking the 36 nanometer capsule.
Yeah, some myosin that's way over here.
From that on, how it moves ahead.
From that, how does it move? Well, think of it as a rubber band, and all these myosins are generating tension and stretching the rubber band, and it's keeping a constant force on the two Z lines, and that force makes them come together. And so as the Z lines move together, the actins are all working to keep tension on the thin filaments, and they keep coming together. Think of it as a cooperative machine rather than as a single motor. All of us axonal transport people like to think about processive motors, right? I'm just trying to point out that processivity is special, and it's important. And frankly, I don't really care anymore about myosin, and I'm going to stop talking about it. Oh, okay, Sandra's question. How does an organelle myosin, like myosin 5, how does it do this? Well, what do you see here? What did it do? What did evolution do? It's got a long neck. And so when it does, at the distance of 2 light chains right here, if this swings, let's see which way are we going? Okay, right here. If this swings 5 nanometers, how far does this swing? How far does it swing? Come on. Double? Well, double, let's see. No, not double. Seven times. 36 it has to step 36 nanometers, right? So this stilt, I don't know if you know what a stilt is. This long, stiff extension lever arm is elongated long enough that now when this thing swings, it can actually make the 36 nanometer jump so that it doesn't have to follow the helical pitch of the actin filament.
Actually, going to the next one, which is 36 into 2.
36 is 2?
36 into 2 of it.
Okay, okay, I was wrong. Thank you, Ruth. So it doesn't have to go 36?
No, it has to go twice this, because this other one is bound at the next one already, right?
It's 36, let's see.
What is the difference between those two heads?
36. Yeah, this one takes a 36 nanometer step. I know that. I didn't measure it, but somebody else did, and they told me. It moves 36 nanometers, and if you want to see that that's really true, you can watch this movie, this amazing movie that Kodera and his colleagues made with an ultra-high-speed atomic force microscope looking at actin filaments with myosin 5 on those actin filaments. You guys should really go look at this paper. It's extraordinary. Isn't that amazing? This is what we dream about, is seeing our motors work.
It's possible because myosin takes such a step to use an airplane to see it.
Because it's so slow? Yeah, I think they use some high viscosity things and various, and low ATP and various things to slow it down.
And they also did reconstruction. So they didn't necessarily take a continuous film all the time. So they did reconstructions.
They cheated? Well, whatever they did, I believe it, and this looks very cool.
There's an important question. Why don't we use microfilaments for transport in accidents? Yeah, the plaques.
Why?
Very obvious answer, but you need to know what it is.
Why are actin filaments not so useful for long distance axonal, vesicle, or mitochondrial transport, fast transport?
Because in neurons, we are having more microtubules than actin.
Well, that's a practical statement of the way things are, but
There's a lot of
theoretically,
It's the length of actin
Dynasty of the microtubules. Microtubules more than.
maybe diamonds that don't yield carbo.
they're not very load bearing. So this is a good point. This is
Does the cardiac, does the motor really have to bear the load of the carbo? Isn't the viscosity of the
the amount of force that's generated by these motors is so much in excess of the mass that's being moved.
or the drag.
You don't have to worry about it. There's no drag to speak of. It's basically a lossless, a loadless issue. Now, there's a much simpler answer. Are there sarcomeres in neurons?
Well,
how long are the actin microfilaments?
Neurons are very large and very long neurons.
So length of actin filaments are shorter than
actin filaments in most situations are quite short.
Very short and they're branching.
They branch. They're good for making a meshwork that has good dynamic strength, but it's dynamic strength, so it can be deformed. But they're not so good for making a long, stiff. So a microtubule, it's hollow, right? That's why it's stiff. If it weren't hollow, it would be smaller diameter. It would be more flexy. And so the stiff microtubule is good for bearing compressive force. You can push things on it. You can put compressive force on it, and it won't just flop over. An actin filament, you push on it, and it just goes, unless it's very short, and unless it's in a meshwork.
Or it's in a sarcomere with all the troponin and tropomyosin.
Right.
And anything else.
Right. It has to be engineered to be an effective long-distance motion machine.
So let me ask a question, right? So when you come up against the plasma membrane, there is a lot of actin right under the plasma
The
membrane.
cortex.
Yeah. And so the final step before you get a vesicle, say exocytosine or some sort of any generic cell, is going to be acting. How do you maintain directionality towards the plasma membrane in that situation?
Well, it's because at a synapse where you don't want to take a lot of time to release your vesicle to get them, well, then they're docked right up against the membrane, so it just takes a little pulse of calcium and bam, they're released. And so it's an interesting question is if you have a 10, say 5 micron thick cortex between the plasma membrane and where the vesicle is, how do you get the vesicle through effectively? And a lot of it's because there is a cooperative pull. There's a bias in actin organization so that the plus ends are more often toward the membrane than away. And there's also a dynamicity to the actin crosslinks in there so that they're constantly forming and breaking. And so if you move things slowly, it's as if the crosslinks aren't there. You just slip into the holes that are made by the dynamic.
Remember, many cells are less than 5 microns.
Yeah, I'm thinking about Microsoft oocyte. Okay, 1 micron.
No, it's not even that. It's much less. But
Really?
The plasma membranes have a skeleton. It's a rather thin element, so you don't have to go very far.
Well, the red blood cell, yeah, the classic spectrum network.
Further, so in case of actin, does it provide bidirectionality? For example, in case of axon, you would need cargo to go in both directions.
Yeah.
But if it's an actin filament, you can't really control that traffic, right?
Yes, you can.
No, but the same motor would be going in both directions. So how do you make sure it is?
So when you have a disorganized, if you have a meshwork, how do you control direction? That's a very interesting question. And what I was trying to push you into thinking was that a bias in the meshwork will do that, right? And so you'll have a random walk around in the network, but if you have say, 60% of the plus ends are toward the plasma membrane and 40% are in other directions, then eventually you're going to go to the plasma membrane. So it's a biased random walk.
If you look at the nucleation of microfilaments at the membrane, they're always with plus end towards the membrane. So that's why, even if it's a meshwork, it's sort of like almost got arrows. So the individual filaments are all sort of moving towards there. Now, because the cell is like this, there's many different directions, but at any given microscopic point, it's all pointing towards the membrane.
So there is bias in the orientation of the polarity, but how we generate the bias in the motion of the actin-based motors at a time when we need both transport in both the directions. Then how will
you don't typically get bidirectional movement of myosins. And there's some special cases where it's been reported, although it's not clear how common that is in vivo. But myosins almost all tend to go towards the plus end. Okay, and so it's the polarity of the actin microfilament that's the critical issue.
Can a carbolus myosin still bind to the filament and work on it?
Yes.
Then not to waste lot of time.
Oh, you mean in a cell? In a cell, can a carbolus myosin bind the filament and do things? I imagine that it can. I mean, if you think about it as a biochemist, everything's in a sort of a thermodynamic equilibrium, and there's a certain probability that you'll have any one binding interaction in force at a given instant. And so very rarely will you have an unloaded myosin bind an actin filament, but it can. It's just not very important because it's rare.
Are there any autoinhibition mechanisms for
Autoinhibition for the cytoplasmic myosins, I do not know the answer to that.
Remember, many of them have calcium-mediated switches.
Yeah.
For instance, on the myosin 5 up there, those light chains, those are mostly calmodulin. So those are a whole series of calmodulins. So depending upon how much calcium is bound to those, you get a difference in terms of the activity. So these motors are not just turned on constitutively, they're regulated, just like kinesin is regulated.
And the light chains can be phosphorylated, and that changes the way they behave too.
Considering the Myosin fiber, it takes the 36 nanometer of step in 180 hydrolysis, and on the another hand, the kinesin takes 8 nanometer of step on hydrolysis of 180. The energy which is provided for the power stroke is the same, but they are taking different, they are traveling different distances. So the power, the force generated during the power stroke will vary according to the
That's good.
conservation of energy.
Yeah, I mean, that's basic Newtonian physics, right? You have a long lever, it can move further, but it's not as forceful.
Yeah, we can say it's a higher gear.
Yeah, it's geared high. Good.
Kinesins are one of the stronger motors out there.
They're geared low. Okay, can we move on now? And how much time did we use up so far, Sandra?
Don't worry.
Well, Roop is going to talk some serious biophysics.
Maybe you can have another 15 minutes.
15 minutes? Okay. Okay, now we're going to talk about the kinesin 1 motor domain, all right, which is sort of probably pretty accurate for most of the filament walking plus end directed kinesins. There it is. That's what we know. No, that's not true. There, that's what we know. More semi-heroic, heroic crystallization structural studies have generated, oh, this was where I was going to draw on the board. And I think it's kind of important so that you'll understand. Now, you tell me if you can't see this, all right? So I have a cheat sheet here. Let's see here. Okay. 1, 2, 3, 4. So this is an alpha helix in kinesin. It is alpha helix number 1. Alpha helix number 2 goes 1, 2, 3, 4, 5. And number 3 goes 1, 2, 3, 4, 5, 6. All right? So that's what we're looking at right here. These purple helices, 1, 2, 3. All right? So that's close to you. Deeper, you have a beta sheet. You can see that as the light blue arrows in there, and you have 1. These are parallel in the beginning. 3. Well, I should have made that one shorter. I'm getting shorter too. Yes. And number 4 comes down here. Number 5 goes the other way. And number 6 goes like that. All right, so that's in the middle. And then on the back side, you have 3, you have 4. So this is alpha 6, this is alpha 4, and then there's a little short one that is alpha 5. And so that's what you see back here. Alpha 5 is green, alpha 4 is green, alpha 6 they made purple again. The green is signifying things that bind to the microtubule. All right? I just wanted to make sure that you understood the layering here, because these structures can be very confusing. And it'll probably be confusing, but that's okay. All right. Can we turn off that light a little bit again? And we'll go to how You can control the lights from here?
No, I just want to turn the camera once. Focus on the board.
Oh, I see, I see. Makes me look really very large. So an important, so we're going to start talking, a number of things here. Okay, so we're looking at what they call a beta sandwich, 3 alpha helices, a beta sheet, 3 alpha helices. Up here in this little depression, there's this short part of the beta sheet, and there's a cluster of these loops and pieces of helices. That's where the ATP binding pocket is, the nucleotide binding pocket. The green parts are what interact with the microtubule. The microtubule is behind the screen. And then down here is where you go off to the tail and the cargo. All right? This is called the neck. That's where kinesin heavy chain of kinesin 1 can first start to dimerize. And then it dimerizes in a long coil going out there. And the force transducer, so myosin has the lever arm, right? Kinesin 1 has a neck linker. So it's basically a zipper that can bind along the side of the head. And that's the force-producing conformational change. All right? And so now I'm going to spin this. I think I'm going to spin it. Oh, yeah, I am. So I just rotated it 90 degrees, anti-clockwise. Okay, so it's the same structure, and you can't read the symbols, but that doesn't matter, because what it is it's just context to look at this simple cartoon. This is from some beautiful experiments done by Sarah Rice. She was able to put different fluorescent adducts on this point and on various other points and show that this neck linker during the ATP ACE cycle, that neck linker swings and indeed binds along the head over here. And so what they saw was that this is just a single kinesin head, to make it simple. This green dot is the fluorophore, and the neck linker here, when the kinesin binds ATP, that neck linker sticks to the side of the head. And so that moves this part towards the plus end of the microtubule. And then you have ADP after hydrolysis, and then it releases. Dimeric head, you can think about it as, okay, now we have to coordinate 2 ATP hydrolysis cycles and filament binding cycles. And so you have the free head has ADP bond. Okay, so kinesin heavy chain is different from myosin, right? When it has ATP bound, it's bound to the filament. Okay, so now we have ADP bound and it's free. This is diffusing around, looking for a binding site. Bang, it hits the binding site. And that triggers release of the ADP. And it can then bind tighter. And then when, let's see now, which one is which here? I get confused at this point. Binding, okay, and so you have hydrolysis then on the back head. And you have so ADP and inorganic phosphate. And there is then ATP comes in and binds to this head, and that is what causes the neck linker to zip along the side of the head. I'm not doing a very good job of this. But let's just go on anyway and look at it. And the dynamic movie. This again is a conceptual cartoon. There's some structural information here, but it's not perfect. ADP release, ATP binds, that causes this neck linker to zip along the side of the head, pulls that back head forward, so that it can hit the next binding site. In this case, the next binding site can be the next tubulin dimer, so it's only 8 nanometer steps, right? Does anybody want to talk about this?
All the chemicals take the same
I think so, but I'm not sure. It's 8 nanometers on the one that's been tested, but I'm always kind of shy about saying something
There are
categorical.
some kinesins which don't seem to move very much.
Don't step
They
at all.
generate force, and so it's a little bit, it's not clear what their step size is.
Yeah.
You know, so, and then there are others that go to the ends of the microtubules and then break down the microtubule. So it's a basic machinery has been adapted for several different purposes.
Yeah, I think if you wanted to have a longer step here, you'd have to have a head and a neck linker that were longer, and it doesn't make
It's probably
sense.
worth noting that the overall topography of the nucleotide binding portions of the myosin and the kinesin are very similar.
Yeah, I didn't say that.
But the neck linker and the way it's adapted to movements are very different.
Could you yell at me, please? It's not rude, it's just helpful. Interesting question. Which came first, kinesin or myosin? I would argue for kinesin.
They're probably an example of convergent evolution, however, not
Really?
one derived from the other. I don't think, there's no sequence homology. It's just structural homology.
Structural homology.
So it's the basic topography of a nucleotide binding site. There are certain ways in which it's very efficient to bind nucleotides for proteins. And so that basic structure came before either one of them, because they're nucleotide binding proteins of E. coli and even viruses. And those were precursors to all nucleotide binding proteins.
So as the G proteins,
Yeah. So
G proteins give the full.
It really essentially originated probably as a switch rather than as a motor producing, and then they got attached to that. And it was probably they branched off very early from something that wasn't. It's not like the kinesin gave rise to the myosin or anything of that sort. So it's probably branching.
Excellent. I imagine in some form, yes.
I mean, the sequence homology is so limited, so it's hard to say at that point because they diverge very early.
All right, I'm trying to prove. 1 minute? I have 1 minute.
One question.
Oh, one question.
You said in the beginning, and there are certainly good examples of linearization of the filament itself acting like a
No motor?
So, apart from cell division, constriction, of course, what are the examples in neurons where this kind of polymerization itself generates force?
Scott, have you ever looked at actin rockets behind vesicles and axoplasm?
Never seen any.
Did you look?
No, we haven't looked, but I don't think anybody's ever reported that. That kind of motility basically has only been reported with pathogens, as far as I know.
Well,
But
there's
you have the movements of growth cones and filopodia generation.
Yeah.
That's clearly
But so that's actin driven.
what he's referring to is that certain viruses and small bacteria that can go inside the cell and they can actually use the polymerization kinetics mechanisms of actin to move around the cell. But I don't know of any examples of that being done normally for vesicle.
In cultured cells, there are a few reports of rockets behind vesicles.
Okay, I have not seen this, so I don't really have any strong feelings.
Basically, if a cell can it's done all the experiments. If it's useful, it will keep it. So I imagine that they're at some level rocketing organelles is fine.
Yeah, it's not a very typical mechanism, but it certainly could exist.
So treadmilling might be known that it somehow is formed from migration of these vertical.
Treadmilling? Treadmilling could work too.
But treadmilling, for the most part, treadmilling does not occur in situ. That's an in vitro artifact, giving exactly the right concentrations of them without microtubule associated proteins, without filtration of post-translational life. All of these things dramatically impact the.
Go ahead.
In that video which you showed, like the stock is not stretching up. It's not like it's upward. So that may decide state size. So how big is it very straight or
You mean the neck? The neck, kinesin has a very short neck. Not like
it doesn't have side enough?
I couldn't quite hear. Did he answer your question?
Asking is why the neck in that movie was shown as twisted and so forth.
Well, it's a random coil, and so it can freely rotate because it's only a single peptide bond, and it can rotate around that bond at any amino acid, right?
Don't take these cartoons too literally.
Yeah. I mean, yeah, and don't take the cartoons literally. I think conceptually it's important for thinking about how you link the filament binding cycle to the conformational change cycle in ATPase.
State size will depend on the size of that.
Yeah, on the size, on the length of the head, and on the length of the linker that lies alongside of the head. That defines how much displacement you can get out of that kind of a machine. I don't think anyone has tried that. You can increase the stepping rate with different engineering tricks, but step size I've never heard of engineering to get that to happen.
In principle, it's possible.
I would imagine some nano guy could build one.
Similar to earlier question of Myosin, the asymmetry for the unidirectional motion, is it built into the motor itself? The structure of the motor has some asymmetry so that it moves only in one direction?
Yes. So the filament has a structural polarity, right? And the charges and the lack of charges, the topography, the binding topography of the filament has to match exactly with the opposing topology of the motor. And since it's got such identity to each one, they can only lock together in one way. They cannot bind randomly. Even though we draw them as spheres and things like that, they aren't spheres. They have tremendous surface topography in terms of their binding interactions. So it's a lock and key, and they can only go in that one direction.
So just that or when the stop is one that is taken from is there a particular direction for the motion?
There is a particular direction because, I mean, if the plus end of the microtubule is down in the floor, this neck linker needs to bind to specific places along the side of the head. Those binding interactions are highly specific between different amino acid side chains and different groups in the polypeptides. And so it goes like this, and this, and this. And it won't bind over here, and it won't bind up here, it won't bind anywhere else. It's another lock and key interaction.
The crystal structure of the dimer shows that they're not symmetrical when they're locked together. So they're not in exactly the same angles. So they're twisting.
And one motor domain is bound to the microtubule filament, and another motor domain is searching for the binding site. That searching is simple diffusion-like motion, or it is also directional?
Well, it's there's about a 4 nanometer, 5 nanometer displacement because of the zipper, right? 4 or 5 nanometers, and then the trailing head. So you thrust this forward. The trailing head now cannot find it gets pulled off and it can't find its own binding site. It's displaced away from it. So it diffuses around, and the next closest binding site is in front. And so it doesn't have to be the full 8 nanometers here. It just has to be far enough to get this thing to not very often rebind to its old binding site and to bias it towards binding towards the forward binding site.
But it stays in the same protein.
Yes.
Doesn't go left or right from that direction.
Rarely, if ever. And it's probably because of, again, the lock and key. If you get some different geometry, you're not going to have a high probability of binding off your own protofilm.
It's a kind of bias around it.
It's a bias, sure. I mean, everything is biased Brownwood motion, right? That's what ATP does. Would you restate that? Say it again.
What about the structural properties of the motor proteins which are bidirectional in nature?
What motor protein is that?
That can move in both the plus direction and the one in the direction.
What motor?
During the movement, it can either go in positive plus.
There are some reports about that, but there are controversies regarding.
Yeah, it's unlikely that motors can move the wrong way, but it's very weak and it's slow and it doesn't work. If you want to move the opposite direction of the same filament, you have to use a different motor. Yeah, so you switch motors. Okay, so I don't have enough time to get into. Are you going to kick me out now? 5 minutes. Okay, so what I was showing you here was sort of the structure of the microtubule and the kinesin. This is viewing from the side of the microtubule. The plus end is this way. And this is, if you turn that around, now if you look from the minus end toward the plus end, you're looking at the back end of the kinesin head, okay? And the point here is this helix, which is alpha helix number 4, is thought to be very important for the mechanism of coupling the filament binding and the ATPase cycles and getting the conformational change. And there's just this little cartoon. This is free of the microtubule. It's not found in the microtubule. Alpha 4 helix, which is also, Sindelar is the guy who wrote this review and did a bunch of work that has recently brought up this hypothesis. Alpha helix number 4 is short, all right? This is loop number 11, it's long. Loop number 11 goes up to a part that senses the presence of the gamma phosphate, okay? And so this loop is what communicates between the microtubule binding event and the ATP hydrolysis event, or ATP binding event. All right, so this then comes and binds the microtubule. And what happens that I find quite interesting is that this loop 11, part of it joins alpha helix number 4, so that the helix gets long, the loop gets shorter, and this lengthening of the helix gives an extra strong binding point to the microtubule. And now, when ATP comes in and binds, all right, ATP sucks this cavity into the main part of the head, which is what this blue thing. This is most of the kinesin head up here now, okay? But it makes this cavity, and this isoleucine number 254 side chain fits right in that cavity, and it causes the head to tilt relative to the alpha 4 switch 2 helix. And that opens up a slot right here into which this neck linker can bind. So it exposes the pocket, so it exposes the pocket and then the neck linker can bind, and that's the way it's coupled. This is a hypothesis. I like it. It probably has a lot of truth and maybe some inaccuracy. All right. This is if you want to think about how the two heads coordinate. This is interesting. I suggest you guys go read this review by Sindelar, Biophysical Reviews. Now, there's a bunch of papers that were assigned to you guys to read for preparing for my talks and stuff. I didn't pick those papers. They really don't have much to do with this. So go read this review and some of the other ones that have been on these slides. All right. Now, I just want to give you one brief look at dining. This is recent, and I'm just learning this myself. The dining has been a terrible protein for people to work on. Actually, they're wonderful on this. But it's huge, difficult for biochemistry, very difficult for structure. But people have succeeded in getting atomic crystals with a resolution for the atomic structure of about 6 angstroms. That's not enough to really see details, but it's enough to see what the secondary structures are. And that's what you see over here. Okay, so you can see alpha helix, beta strand, stuff like that. You can't see exactly how they interact, but you get the general idea. So dynein has 6 repetitions of a domain, AAA ATPase domain. Okay? Yeah, this is very different from myosin kinesin, different fold. AAA ATPase, as opposed to the G protein related ATP binding nature of kinesin and myosin. So the point is, I don't want to go in, I don't have time to do this very long. Here's the microtubule binding site on this extension from the AAA number 4. All right. Now, microtubule binding here has to communicate to the ATP hydrolysis site. Who knows where the ATP, the important ATP hydrolysis site for force production is? Which one? Number 1, right here. Okay. So somehow this way over there has to communicate with this way over here. Interesting problem. And there's plenty of room for people to find the answer. There's some suggestions that this coiled coil here is kind of, it's got 2 different registrations, and it can go like this or like this, depending on whether it's bound to the microtubule. And this slippage right here can operate some structural changes in here, and there's some imagination that this is like an iris, and in one state it'll close up, in another state it'll open up, and that can ripple some conformational changes around here. They're good ideas. They may change. Okay, so on top, so here's the important part right here, all right? This is the linker, and this goes off to the tail and the cargo binding portions of dynein, all right? And it turns out this is probably what wiggles to generate a step. Fine. And here it is. So here's the cargo. Here's the stalk tail equivalent of dynein. Here's the AAA ATPase ring. And here is that linker. And there's the microtubule binding site. And so what happens is ATP binding causes release. Why don't I use the pointer? Causes release from the microtubule. And the binding site, the dynein microtubule binding site, changes from a square into a circle. Scott's laughing because he knows I'm joking. Okay, you guys know I'm joking, right? It makes a conformational change, so it no longer wants to bind the microtubule. All right. So now you added ATP. Boom, you come off the filament. That's like myosin. It's not like Myosin, but it doesn't matter. The linker gets bent. All right. You have microtubule binding, and then that causes the linker to straighten out. And so you can cause a change in distance between the cargo and the binding site on the microtubule of presumably about 8 nanometers total, or 4 or 5 nanometers, so that diffusion can find the next one. Dyning is not so good at tracking on a single protofilament because of this huge head. It has to kind of waddle, and I think it can. Some people think that it actually walks down adjacent protofilaments, or one way over here. It has a lot of flexibility. Some people think that it walks on a single protofilament. I don't have an opinion. I'm just reporting what I've read.
There was a while ago. There was an EM-based structure.
That's what this comes from.
Yeah.
Yeah.
And did they look at the same order and the same amount, same region of the protein? Is the crystal structure and these legs match them?
The sequence similarity is quite strong between the different dynein heads. I mean, yes. And the electron microscopy done by Roberts and colleagues was done with both cytoplasmic dynein and with the C subunit of flagellar dynein, and they're similar. And what they see that's dramatic is this bending of the linker, and that I think is almost certain to end up being the important conformational change that causes displacement of the head of the cargo toward the minus end of the microtubule. So I think I'm getting tired. Are you guys getting tired? If you're really tired, then think about this. Go back to axoplasm, go back to reality, and think about how all of these motors link to their cargoes, how the motors relate to one another or don't relate to one another. Are they separate? Are they together? How is it that they don't have a tug of war, except in rare instances? They somehow have to coordinate with one another, and they have to be regulated to drop this mitochondrion off where it needs to be, which those are the things that Scott's been talking about, was talking about earlier with the regulatory schemes. There's all kinds of, there's just a rich set of problems here to solve, for you guys to solve. Are you hungry? We can have questions at lunch or something.
Yes, I think in the interest of time, we should thank Bill.
Ruth, I'm sorry.
Okay.
So the room is
Maybe we can have lunch.
Lunch is still about
20 minutes?
1 hour.
1 hour away. Well, then you do have time. Should we have a break or not?
I think we can have like maybe a 5-minute break, where just water break, and then we can stop.